All right, welcome back. So we're going to be finishing up the hepatitis chapter here, and the last section to cover is the molecular virology. And as you might anticipate, this can be a lot because there's so many hepatitis viruses, and they're all distinct viruses, although there is some similarities between some of them, as we'll discuss. But I'm going to try and keep this as simple as possible. I'm only going to cover hepatitis A, B, and C because D and E are very uncommon or unknown in the US. So we'll focus on the three that you do find in the US. And you don't have to memorize the names of like every protein encoded for by each of these three viruses. So I'll try to give you the overview, and like I said, keep it as simple as I can here. Another good thing is there are going to be a lot of similarities between hepatitis A and hepatitis C. So we'll go through these in order, but just keep in mind as we're going through hepatitis A that pretty much everything we discuss there is going to come back around in hepatitis C. Hepatitis B is going to be its own animal for the most part. So, as I mentioned, all these viruses, even though they all cause the same clinical disease, they are distinct viruses and, in fact, get classified in completely different families. You do have both hepatitis A and C being positive sense single-stranded RNA viruses. Hepatitis B is something actually quite unusual that we'll get into here in a minute. They've all got small genome sizes. They're all small icosahedrons and varying numbers of genotypes or subtypes, subspecies of virus, depending on how you want to think of them, strains, if you will. So, like I said, we'll go through them in order. We'll start with hepatitis A here. This is the only one we're going to talk about that's a non-enveloped icosahedron, and this one is the smallest of the three. In the Picornaviridae family, which also includes poliovirus that we talk about this semester. There's just one serotype of HIV, although there are 4 different genotypes. So what this means is even though there's 4 distinct strains of the virus with differing genomic sequences, the antibodies that would be produced against them will recognize all 4 of those genotypes. That's what serotype is referring to. So that's a good thing, right? You wouldn't have to worry about getting infected with all 4 to have full protection. So in terms of the genome organization and the way the genome is transcribed and translated, as mentioned, this is a positive sense single-stranded RNA genome, about 7.5 kilobases, and those positive sense RNA genomes, remember, can basically function as messenger RNA, so they can be directly immediately translated in the host cell without any additional modification. And you may remember messenger RNAs, they always have a 5' cap and a 3' poly A tail. So if our viral genome is going to function as a messenger RNA, it needs those as well. And you can see it does have a short poly A tail at the 3' end, at the 5' end. It's got the NTR, non-translated region, with this what's called the iris, which is internal ribosome entry site, and it's got this secondary structure that basically mimics the 5' guanosine cap and is going to allow the ribosome to recognize this as a RNA that's ready for translation. And interestingly, what we see is just a single open reading frame. So the whole genome basically is just going to be translated as one polyprotein, And we can see here, just in terms of functionality, that's basically been divided up into 3 sections, P1, P2, P3, where P1 is basically going to form the capsid proteins, P2, proteins important for replication, and then P3, you've got like the RNA polymerase, the RDRP, as well as protease, and a couple of other players here as well, the VPG, which is what we see attached to the 5' end up here. But as I was saying, this will get translated into one long polypeptide, and then proteases are going to cleave that into all these individual proteins. Right? So this is pretty common. We'll see this come back with hepatitis C, but also with other viruses this semester. And like I was saying, you don't have to memorize all of these proteins and their functions. So I won't get into that at this point. So then in terms of kind of the overall replication cycle, of course, we start that with attachment and penetration. And hepatitis A can infect both T cells and, of course, liver cells, hepatocytes. So it's got different receptors for those 2 cell types on T cells called TIM1. It's this long name, T cell immunoglobulin and mucin domain 1, versus on the liver cells, it's ASGPR. We'll just stick with the abbreviation there. So we get that binding at the cell surface. I don't believe it's known exactly what on the virion is interacting with those receptors, but clearly there's some interaction there, presumably maybe with the capsid protein itself. And then it has been demonstrated this virus gets taken in by non-clathrin mediated endocytosis, is then going to end up in this endosome or endocytic vesicle, and you're going to get fusion and uncoating there, which may be mediated by the VP4 protein. Let me just jump back. That's this guy here, the very end. But there's a question mark next to that, so I think that's still a little unresolved in the literature. But anyway, I think they are pretty confident this is where uncoating happens. And then that is going to have the effect of releasing that positive sense single-stranded RNA genome into the cytoplasm. Of course, there's going to be ribosomes there, so we can start to get immediate translation. Right? That's what they're showing happening over here. And again, we make that polyprotein, cleave it into the individual viral proteins. One of those, of course, one of those viral proteins is going to be the RDRP, the RNA polymerase. And once that is produced, we can start replicating, making additional copies of the viral genome that we're going to need to produce new virions. And so the way this goes is we will take our positive sense RNA genome, make a negative sense, what's called the anti-genome copy, right? So the RDRP does that, and then the RDRP will also take that and then convert it back into the complementary positive sense copy. So we do a little dance there, but we end up back where we started, basically, with the more positive sense copies. Once we produce capsid proteins and those positive sense copies, then we can form new virions. And again, we got a question mark here in terms of how the virions are getting out, but they're suggesting possibly through exocytosis, but they don't seem to know a lot of the details on this guy just yet. So pretty simple. Honestly, that's about as simple as a viral replication cycle can get. And good news is, as I said at the beginning, we're going to see something pretty similar with hepatitis C here coming up. Hepatitis B, however, more complicated for sure, and part of that comes down to its unique genome, which is this partially single-stranded, partially double-stranded DNA genome, as you see here, right? So this part single-stranded on the right, good chunk on the left here, and up top, double-stranded. But before we get into that, again, this is icosahedral virus. This one is enveloped, and you can see you have these three different flavors of glycoproteins on the surface: HBS, AG, L, S, and M. You don't have to memorize those names. But it's got a very small genome that has that unique structure we were just talking about. They've identified 8 different genotypes, so hepatitis B, A through H. We find all 8 of those circulating in the United States. And again, there's those 3 different glycoproteins mentioned again. But one thing that's very unique with hepatitis B is you actually in infected cells will form 3 different types of particles, only one of which is infectious. So that's obviously the one we're going to focus on, is what's called the Dane particle here. But you also end up with these small spherical particles, as well as these kind of elongated football-shaped filamentous particles, they're called. And you can see those on the micrograph here. So here's kind of several Dane particles in the middle. You can see the longer filamentous and the little spherical particles in the background. And, neither of those smaller particles contain the genome, which is obviously going to contribute to them not being infectious. But what's kind of interesting, and I don't think totally understood, is why these are being produced. And in fact, they're actually way outnumbering the infectious particles in an infected cell. They estimate there could be as many as 10,000 times more of these small non-infectious particles than the Dane particles. So weird for sure. So we'll go through the replication cycle in pieces, but before we do, I just wanted to kind of give you the 10,000-foot view, and then we'll jump into some of these in just a little bit more detail. Like I said, we're not going to go crazy here, but of course you got attachment at the cell surface, right? We're going to get into the cell by clathrin-mediated endocytosis. There's going to be a fusion event with that endocytic vesicle that is going to allow the nucleocapsid to escape. That is going to go dock with a nuclear pore complex, and this is where uncoating is going to happen at the nuclear pore, and that unusual genome that we discussed is going to be injected through the nuclear pore into the nucleus. And again, we've talked about this repeatedly this semester, but if you're a virus with a DNA genome, you most likely need to gain access to the nucleus, because that's where the polymerases, the enzymes you need for transcription and replication, are going to be found. So we get our, we mess with the genome a little bit. We'll get into this in a second. Inside the nucleus, we get transcription of a few different RNA types. And of course, those are going to be translated out in the cytoplasm. But at some point, we're going to package up what's called our pre-genomic RNA. That's going to actually mature here inside the cell through this reverse transcription step. And then in terms of getting out of the cell, because this was an enveloped virus, the virion's actually going to bud through the ER, pick up 2 layers of membrane, the outer layer of which is going to be discarded as it buds out of the cell by exocytosis. So let's jump in and just look at some of those pieces individually here. So attachment, we do know what the receptor is, this NTCP protein, and we do know that interaction is going to be mediated by at least one of those glycoproteins, the L-glycoprotein on the surface of the viral envelope. As we said, that's going to trigger clathrin-mediated endocytosis. and there's going to be a fusion event here between the proteins on the surface of the viral envelope and the membrane of the endocytic vesicle. That's going to get the virion out, where it's going to be transported to the nuclear pore, and then the DNA will be injected into the nucleus. So we had this unusual partially single-stranded, partially double-stranded DNA genome structure that is referred to as rcDNA, which stands for relaxed circular DNA. And this is going to be quote-unquote repaired in the nucleus into what's called cccDNA, standing for covalently closed circular DNA. So basically, in a nutshell, we're going to make this fully double-stranded, right, circular DNA genome inside the nucleus, using host polymerases to accomplish that. Excuse me. Had to take a little drink there. So then once we have our cccDNA, that can be transcribed by host RNA polymerase 2 into 3, and I'm going to argue really it's 4 viral messenger RNAs, but we'll stick with the book, 3. And I'm not worried about this for the test, but each one of those 3 encodes different proteins that you can see listed over here on the left side of the slide. I will just point out the third of these messenger RNAs encodes this protein that can directly trigger cell proliferation and is linked directly to induction of liver cancer by hepatitis B. So that one's kind of interesting. But we have these messenger RNAs. Obviously, if you remember your basic biology, right, those are having to leave the nucleus, getting translated by ribosomes out in the cytoplasm. And I said I would argue there's really 4 messenger RNAs because we are also going to create this pregenomic RNA, pgRNA that you see here. Kind of has this little hairpin loop in it that we'll revisit here in a minute. But this also functions as a messenger RNA to create a couple of proteins, in particular reverse transcriptase and nucleocapsid protein, as well as serving as the template for basically creating copies of the HBV genome through reverse transcription, which is what we're going to look at next. So here's our pregenomic RNA, right? Actually leaves the nucleus. It gets packaged up by the capsid protein to make our nucleocapsid, but obviously this is not the genome form that we want to have in our virions, so we need to convert that into the partially single, partially double-stranded circular genome. And as I mentioned, we're going to do that through reverse transcription. So that's actually going to happen out in the cytoplasm within the virion, that reverse transcription step. So obviously we would have had to have also packaged up the reverse transcriptase protein, viral protein that was being produced. And I don't want to get lost in the weeds here in terms of reverse transcription. This was pulled from chapter 4, right, where we covered the nucleic acid replication of all the different types. But I did just want to point out, and this is going to be true of almost any reverse transcription, you need some kind of priming to get things started. In this case, our hairpin loop is going to serve that purpose. So the reverse transcriptase, excuse me, which is the P protein, is going to bind to that hairpin loop and be able to extend off of it. And another thing that you'll see commonly with a lot of these kind of more complicated replication models is you'll have sequences within the pregenome here, if we want to stick with that nomenclature, that are repeated, right? So you see this little DR1 sequence found kind of at both ends of the the genome here. And what this is going to do is it's going to allow a partially reverse transcribed copy now to jump from one end to the other because of the sequence similarity there. So we see this jumping around. Again, that happens very commonly. Once we're over here at the 3' end, now we can extend basically make the full genome reverse transcription at that point. And then we do another little dance in this case to make this thing partially double-stranded, right? Kind of coil it all up, and we're basically there. Obviously, that's an oversimplification, but gives you the basic gist of it. So then we're really ready for prime time. We've got our nucleocapsid with the appropriate genome. We want to get this thing out of the cell and enveloped. And this is a very common strategy you'll see in quite a few viruses where, quite a few envelope viruses, I should say, where we're going to bud through, say, the ER or the Golgi to acquire that membrane. So what you can see here is the glycoproteins, right, that we find in the viral envelope that were being produced in this infected cell have already localized themselves to at least a portion of the endoplasmic reticulum, the ER membrane there. The virion is going to be attracted to that portion of the ER. It will, bud into the ER, acquiring its envelope with all the appropriate glycoproteins as it goes in. We don't want to leave it in the ER, so it's going to bud back out, and now it picks up a quote-unquote normal ER membrane, a second membrane that it doesn't want or need, but it's going to lose that when that outer membrane fuses with the plasma membrane here during exocytosis. So what's actually released is our fully mature enveloped virion. And then that's just that 10,000 foot view again, if you want to refer back to it. Okay, so then that leaves us with hepatitis C, which, as I said, because of its positive single-stranded RNA genome, is going to have a lot of similarities to hepatitis A. I think that's probably a relief at this point, right? Because hepatitis A wasn't too bad. This one is going to be enveloped, another icosahedral virus. Different family again, right? Almost 10,000 nucleotides in the genome, which is still quite small. We've got 11 major genotypes of hepatitis C that have been identified, and they actually have quite a bit of diversity. That level, more than 30% diversity, is probably going to mean that this is like multiple serotypes in terms of immune recognition. And you'll notice in the viral envelope, you have these E1 and E2 cell surface proteins there, and as you can probably guess, those are going to be important for attachment of the virus. So I won't dwell on it since there's a lot of similarities here to hepatitis A, right? So we've got the similar 5' cap, internal ribosome entry site at the 5' end. We've got the poly A tail. We're going to translate this in just one long polyprotein, and then proteases are going to cleave that up into the individual functional viral proteins. So at this point, we're pros at that idea. So we do know some things about hepatitis C replication cycle, but I wanted to point out that it has certainly been the most difficult of the hepatitis viruses to study because it just doesn't replicate well in traditional cell culture or mouse models. That's created headaches. So I think researchers have gotten around that by creating some, genetically altered transgenic mice that are basically optimized for being infected with Hep C. Poor mice, but important for our understanding and research. So one thing that's unique and worth pointing out here with Hep C is there's quite a bit of evidence showing that hepatitis C will associate with two different lipoproteins, either VLDL, which stands for very low density lipoprotein, or LDL, just low density, which you might be familiar with if you've ever had kind of your standard blood lipid panel taken. But we think the virus is associating with these guys because they are going to go to the liver, which of course is where the hepatitis virus wants to go, where its receptors are located. So it's kind of using these lipoproteins as a Trojan horse almost, if you will, to at least deliver it to the liver. And I'm not going to test you over these, but I guess I do want you to know there's 4 different receptor proteins that seem to be required for infection. We are interacting with those E1 and E2 envelope proteins, as I mentioned. And we think this virus is then going to be taken in by clathrin-mediated endocytosis, so nothing too out of the ordinary there. Similarly, fusion with that endocytic vesicle that's going to lead to the nucleocapsid getting out into the cytoplasm. And we think in this case the uncoding is happening here in the cytoplasm. And just as we talked about with hepatitis A, once that nucleic acid is out, right, it can be translated and copied. And we're basically ready to package things up and leave the cell. However, one thing that is very interesting and unique with hepatitis C is it's been well documented this virus will actually, the term they use is remodel the ER membrane to create these what are called double membrane vesicles or multi-membrane vesicles. I'll show you a picture in a minute, but essentially it's creating little chambers out of the ER membrane where virus replication is going to take place. And I think it's just an efficiency thing. You actually see this happening in quite a few different viruses, or some version of this happening, where the virus will make a little replication complex or chamber, just able to have all the appropriate enzymes concentrated and in close vicinity to optimize the time frame of that. So here's what that remodeling looks like. You can see like the cartoon illustration of that on the right versus the electron micrograph on the left. But yeah, you can see lots of these little double membrane vesicles, multi-membrane vesicles being formed. So you can imagine the virus genome being in there, and the polymerases needed to make copies there. So we have that happening. Once we've got our viral proteins and our copies of the genome, similar idea to hepatitis A, we've got our RDRP that creates the antigenomic negative sense copy, and that's converted back into positive sense copies. We assemble our capsids, and we're going to see, because this was enveloped, we're going to see budding through the ER, and the E1 and E2, those envelope proteins, right, have already localized themselves to the ER membrane. The capsid is attracted to those proteins or that region. When we bud into the ER, we pick up that envelope with the appropriate proteins. We bud out of the ER, picking up the extra membrane again, and then discarding that upon exocytosis and release. So not too bad. You do have that little interesting addition of the ER remodeling in this particular virus, though. So I think that is it. Yeah.